Hi, this is Master. In this video, we're gonna talk about glycolysis. So let's define glycolysis. Glycolysis is a series of enzymatic reactions which convert a molecule of glucose into two molecules of pyruvate. During this reaction, energy is released in the form of ATP and NADH. Okay, then how these molecules of glucose get in the cell? These molecules of glucose get in the cell through blood transporter. As you see here in the membrane, there is a blood transporter. When these molecules of glucose get in the cell, it has to be immediately phosphorylated. The phosphorylation is done by two types of enzymes according to the type of tissue. This enzyme, it can be hexokinase or glucokinase. Hexokinase is mainly concentrated in muscles, but glucokinase is specific for liver. Okay, so how hexokinase and the glucokinase phosphorylate the molecules of glucose? Hexokinase or glucokinase take one molecule of phosphate from ATP. Okay, so ATP minus one phosphate, it will become ADP. Now the taken phosphate from ATP will be added to the molecules of glucose at carbon number 6. Now the glucose plus phosphate at carbon number 6, it is called glucose 6-phosphate. Now these molecules of glucose 6-phosphate, it undergo the process of isomerization. This process is catalyzed by phosphohexose isomerase. As you know that the isomer of glucose is fructose, then phosphohexose isomerase will convert glucose 6-phosphate into fructose 6-phosphate. Okay, then fructose 6-phosphate. Fructose 6-phosphate is a substrate of phosphofructokinase 1. What phosphofructokinase 1 do to fructose 6-phosphate? Phosphofructokinase 1 will take one phosphate from ATP. Okay, it will take one phosphate from ATP. So ATP minus one phosphate, it will become ADP. Then the taken phosphate from ATP will be added to fructose 6-phosphate at carbon 1, thus converting fructose 6-phosphate into fructose 1,6-bisphosphate. Okay, then fructose 1,6-bisphosphate, it will be split into two halves, producing two molecules. They are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This reaction is catalyzed by an enzyme. This enzyme is called aldolase. Okay, then only glyceraldehyde 3-phosphate will continue the rest of the glycolysis. So as dihydroxyacetone phosphate is an isomer of glyceraldehyde 3-phosphate, both of them can be interconvertible. Dihydroxyacetone phosphate can be converted into glyceraldehyde 3-phosphate, and the glyceraldehyde 3-phosphate can be converted to dihydroxyacetone phosphate. The enzymes which convert dihydroxyacetone phosphate to glyceraldehyde 3-phosphate and convert glyceraldehyde 3-phosphate to dihydroxyacetone phosphate is called triose phosphate isomerase. Why it is so named triose phosphate isomerase? Both of them, dihydroxyacetone phosphate, contain molecules of phosphate, and the glyceraldehyde 3-phosphate contain also a molecule of phosphate. Triose because both of them, they have 3 carbon. Okay, and isomerase because dihydroxyacetone phosphate is an isomer of glyceraldehyde 3-phosphate. Let's continue then. Glyceraldehyde 3-phosphate is a substrate of glyceraldehyde 3-phosphate dehydrogenase. So what glyceraldehyde 3-phosphate dehydrogenase do to glyceraldehyde 3-phosphate? Glyceraldehyde 3-phosphate dehydrogenase, first it is going to oxidize glyceraldehyde 3-phosphate by removing one hydrogen. Okay, the removed hydrogen molecules will be taken up by special hydrogen acceptor that is called NAD. Okay, NAD plus one hydrogen, it will become NADH. Remember that why we have two NAD and two NADH, because we converted dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Because of that, we have two NAD and two NADH. Then the same enzyme glyceraldehyde 3-phosphate dehydrogenase will add inorganic phosphate to glyceraldehyde 3-phosphate at carbon position 1, thus converting glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate. Okay. Then 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate is a substrate of phosphoglycerate kinase. So what phosphoglycerate kinase do to 1,3-bisphosphoglycerate? Phosphoglycerate kinase will take one phosphate, or it will remove one phosphate from 1,3-bisphosphoglycerate. So as we know that kinase will add phosphate, so the removed phosphate from 1,3-bisphosphoglycerate of course will be added to something. Okay, so the removed one phosphate will be added to ADP. So ADP plus one phosphate, it will become ATP, and now we produce ATP. Why we produce two ATP? Because dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. So we have two glyceraldehyde 3-phosphate, then we have two 1,3-bisphosphoglycerate, and now we produce two ATP. Now 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate by the enzyme that is called phosphoglycerate kinase. Okay, then 3-phosphoglycerate. 3-phosphoglycerate undergo a process of mutation. What does mutation mean here? Here that means a special enzyme that is called phosphoglycerate mutase will move this phosphate into position 2, from 3 to 2. Okay, look this animation how phosphate is moved. This function is done by phosphoglycerate mutase, converting 3-phosphoglycerate into 2-phosphoglycerate. 2-phosphoglycerate is a substrate of enolase. So what enolase do to 2-phosphoglycerate? It will add enol. So now 2-phosphoglycerate is converted to phosphoenol pyruvate. Then phosphoenol pyruvate is a substrate of pyruvate kinase. So what does pyruvate kinase do to phosphoenol pyruvate? Okay, remember that any enzyme that contain or having the name kinase, it will add phosphate. So pyruvate kinase will take or remove the phosphate that is present in phosphoenol pyruvate, and it will add it to ADP. Okay, so ADP plus phosphate, it will become ATP, and now we produce 2 ATP. And in the last reaction, or in the past reaction, we produced 2 ATP. So 2 plus 2, we had produced 4 ATP. In the beginning, we had consumed 2 ATP. Okay, then now phosphoenol pyruvate is converted to pyruvate, and the pyruvate is the end product of glycolysis. Of course, we produce 2 pyruvate. Then in presence of oxygen and mitochondria, these molecules of pyruvate will be converted to acetyl-CoA, and that we are going to talk about in the next video. That's about Krebs cycle or citric acid cycle. And when pyruvate is converted to acetyl-CoA in the presence of oxygen and mitochondria, this is said aerobic glycolysis. But in case of absence of oxygen or mitochondria, the pyruvate will be reduced to lactic acid. Okay, so we had produced in the beginning 2 NADH. Now in case of absence of oxygen and mitochondria, the produced NADH will be reoxidized. Okay, reoxidized to NAD, and the pyruvate will be reduced to lactate or lactic acid. Okay, so what is the name of the enzyme which convert pyruvate to lactate. This enzyme is called lactate dehydrogenase. Remember that in the absence of oxygen or mitochondria, pyruvate will be converted to lactic acid or lactate. Okay, then let's see what we generated during the process of glycolysis. Glycolysis, it occurs in the cytoplasm because all the enzymes needed for the glycolysis, they are located in the cytoplasm. So one of the characteristics of glycolysis, it is cytosolic pathway. And then our starting substrate is glucose and our end product is 2-pyruvate. During the process of glycolysis, we produced 4 ATP, and in the beginning we had consumed 2 ATP. So 4 ATP minus 2 ATP equal 2 ATP. So net production of ATP per glycolysis is equal to ATP. And also don't forget your friend, we had produced 2 NADH. In case of aerobic glycolysis, the 2 NADH will be utilized in electron transport chain to produce more ATP. But in case of anaerobic glycolysis or absence of oxygen or lack of mitochondria, this 2 NADH will be utilized to produce lactic acid. I hope you enjoyed the video. Please like the video, subscribe to my channels, and comment in the comment section below, and see you in the next video. Thank you.